Hi everyone, it's Mr. Sinti, and thank you for joining me today. Today I'd like to walk you through a really cool and interesting investigation where you would really be using CRISPR-Cas9 to edit and basically knock out the LacZ gene in E. coli. And so I want to go over a little bit of the background on that, as well as some of the experimental methods involved in this. It's really interesting because it's a bona fide CRISPR modification, and you could verify results of the CRISPR modification by growing bacteria on a variety of agar plates. And so, just a shout out to the biotech company Biorad Laboratories and their Explorer program for providing reagents and protocols for this particular study. And so let me jump right into that. Using CRISPR-Cas9 to edit E. coli. So right out of the gate, I want to remind everyone that CRISPR is an adaptive antiviral immune system discovered in bacteria. And so this is, you can see bacteria growing on this petri dish. And so they've developed the ability to fight off and combat viral infections by phage by using CRISPR-Cas9 gene editing. And here's a picture of some phages that are attaching to bacteria. And this CRISPR investigation is based on the ability of CRISPR to protect them from various viral infections. And so CRISPR itself stands for clustered regularly interspaced short palindromic repeats. And it was first discovered in E. coli back in the late 1980s. And this investigation is also about E. coli. And so in this investigation, CRISPR is going to be used to edit the chromosome of E. coli. So normally E. coli, just a little background, normally E. coli, as you can see growing here on this petri dish, forms these sort of white creamy colonies grown on LB agar. E. coli have a gene, you might be familiar with it, on their chromosome called LacZ, and it codes for a particular crucial enzyme that they have called beta-galactosidase, or just simply BGAL for short. It's the enzyme that catalyzes the breakdown of the sugar lactose, the hydrolysis of the sugar lactose into glucose and galactose. Lactose, you can see here that it's a disaccharide, and it's held together by a 1,4 beta-galactosidase linkage. And so the enzyme BGAL comes in and helps hydrolyze that to produce glucose and galactose. And so normally BGAL, the enzyme, which is produced by the LacZ gene, breaks down lactose. And so BGAL normally breaks down lactose, but it can also break down a substrate called XGAL, which is colorless. As you can see here in this diagram, the disaccharide lactose, as you can see here, and the chemical XGAL mimics lactose, especially in this area right here, the beta 1,4 galactosidase linkage right here. And so the enzyme BGAL will treat XGAL as if it's the substrate lactose. And so it's also capable of breaking down XGAL into two monomers. So BGAL can break down XGAL. And what's cool about that is that XGAL is normally a colorless substrate, but when it's broken down into its two monomer components, it turns blue. And that's really useful because you can see that and it reports the efficiency of XGAL. And so there's the LacZ gene. It's found on the chromosome of bacteria. Again, it is transcribed and translated, and here's the enzyme itself, to this protein. And it's this protein enzyme that's going to hydrolyze or cleave the substrate XGAL, which is synonymous with lactose. And it's going to break it down into blue pigments, and it's going to turn the whole cell blue, and as a consequence, you'll be able to see it very distinctively as blue colonies growing on an LB plate that has XGAL in it. Okay, and so the starter plate in the beginning of this investigation is going to have this chemical called IPTG and xgal. And so IPTG, a little background on that, is it's an inducer which basically mimics lactose in a sense that it is going to induce the expression of the lactose operon. There's a repressor protein that binds normally to an operator sequence in the lactose operon, and when IPTG is provided in the growth medium, this removes the repressor protein, which then basically induces the production of B-gal. And so that's going to be important to have in the starter plate. So the functioning bacteria with a functioning LacZ gene will be blue if you grow them on a media with xgal. So on this particular starter plate, you have LB, which is the typical growth media, and then you have the chemical xgal, and then the inducer IPTG, so that the bacteria will express the xgal operon or gene in the operon so that the colonies will turn blue. And I thought I'd include a diagram here showing a portion of the lactose operon. Here you can see the promoter region where RNA polymerase, which is the enzyme that makes RNA, is going to attach. And then if there's a repressor protein that binds to the operator sequence, it's IPTG that will attach to it and then remove it. So therefore, IPTG induces the expression of the following genes in the operon, which includes the LacZ gene. And if the LacZ gene is present, it's going to produce beta galactosidase, or just B-gal enzyme, and that's going to cleave X-gal, and that's going to turn the colonies blue. And that's in a functioning LacZ gene. So how about this? What happens if the LacZ isn't functional and it's not creating beta galactosidase? You're like, if it's not producing B-gal, let's see. I guess if you grew it on a petri dish with X-gal, it wouldn't be able to break down that dimer, and so therefore the colonies would not turn blue, right? And so predictably, that's exactly what's happening. And so no B-gal, no blue, the colonies will still be able to grow because, as it turns out, lactose isn't required by E. coli to survive. And so they're still able to live, but the colonies would be their normal white color. And that's an example of a phenotype that would indicate a non-functioning LacZ gene. So you can think of, and this is an important point in terms of CRISPR-Cas9 in general. We're working with, in this investigation, the LacZ gene, but you can think of the LacZ gene in E. coli as a model for any gene in any organism that a researcher would want to modify. And so scientists can go in and delete portions of genes, insert DNA into a gene, replace a sequence. And so this is where CRISPR-Cas9 genomic editing becomes really important. But to be able to do it is super cool, because it's one thing talking about it, but to actually going in to E. coli and editing their LacZ gene is I think pretty remarkable. So by using CRISPR, we can target and edit any specific genes. And so in this investigation, we're going to start with E. coli with a functioning LacZ gene. And so again, to review that, those are the blue colonies because they're capable of producing B-gal, which breaks down X-gal. Then we're going to use CRISPR to cut that gene. And in some portions of the investigation, then edit it so that the DNA is non-functional. And so basically, we're going to try to knock out the LacZ gene using CRISPR. And again, if it was knocked out, the colonies would be white in color. And so CRISPR is a great tool for knocking out specific genes. It's used quite frequently in science to investigate the normal functioning of a gene if you didn't know what the gene was doing. But by knocking it out and seeing what the results are phenotypically, you can then backward design and say, well, I guess that's what it was doing, that now that it's not doing this, or what protein it's working on. And so we're going to go in using CRISPR, and we're going to try to make the lactose or the LacZ gene non-functional. And if it's non-functional, it won't be able to produce B-gal, and therefore not break down X-gal, and therefore grow white. And so we often focus when we talk about CRISPR in terms of its ability to cut, and that's incredible. The enzyme nuclease Cas9 is capable of, with an RNA guide, targeting any sequence. In this investigation, it'll be the LacZ gene, but it can go in and make cuts to DNA, and this is how it fights off phages normally as an adaptive immune system. It cuts the phage DNA. But what about the repair? That's really important too. And so I want to bring that up. But first, about the cutting a little bit more. So first, the Cas9 enzyme, as you can see in this diagram here, is the enzyme that's going to be capable of cutting DNA. But how does it find the particular area of cutting? Well, it finds this area by using something called a single guide RNA. And the single guide RNA has an area right here where it's going to be complementary to the target sequence that you want to edit. So this 20 nucleotide base sequence is important for guiding the complex to the location where you want it to cut. This region over here, this sort of scaffolding area over here in the hairpin loop, is helping it to bond to the Cas9 protein in particular. And so the single guide RNA coupled with the Cas9 endonuclease forms a complex that can seek out, and in this investigation, can seek out the lactose Z gene in E. coli, or any gene really that we're interested in, provided that we provide the proper guide sequence. This can go and make some edits. And so together they find the target DNA based on the guiding nucleotide sequence right in here. And so the target DNA in this investigation is LacZ, but they use this sequence right in here. And so what it does is the enzyme, meaning Cas9, it unwinds the complementary sequences, and then it binds to the RNA, and then the enzyme makes a cut. And so the Cas9 makes a double-stranded cut in the DNA, as you can see right in here. And so we're going to cleave the DNA. Now, that's not necessarily a good thing when it comes to bacteria, because sometimes, just in general, if a bacteria happens to have a double-stranded cut in its chromosome, how does it handle that? Well, not well is the short answer. If they don't repair it, it's going to be lethal. They don't like it. So bacteria have evolved ways in which their DNA can be repaired so that they don't die. And so double-stranded breaks in DNA, a bad thing for bacteria. And so bacteria have evolved ways to repair the genes so that they don't have these double-stranded breaks, or otherwise they're going to die. Okay, so I want to talk a little bit about the repair mechanisms. Now, basic discussion of this involves two types. So there's one type of repair that cells use called non-homologous joining. And then there's another type called homology-directed repair, or HDR. This is the particular repair that we're going to be looking at in this investigation. So what happens in homology-directed repair is that when the break is made, instead of having the bacteria just struggling to put in any random nucleotides to seal that break, and that's what non-homologous joining is, and oftentimes that results in a harmful mutation. This time we're going to provide it with our own template DNA that's going to fit perfectly into the cut area. and that's known as homology-directed repair. So the target DNA has been cut. Now the bacteria can use the process of homology-directed repair to fix this double-stranded break. And so this process is done by providing the E. coli with some donor DNA. And here it is shown right in here. And then this donor DNA is basically patched into the cut region. And how it's patched in is that it uses repair enzymes, or repair mechanism, if you will. It uses a set of enzymes to help ligate the template donor DNA into the cut region. And so this is known as homology-directed repair, or HDR. So this is how sometimes researchers can repair a mutant gene with a healthy sequence, because you can sort of replace this with this template right in here. You can replace a defective area. And in some cases, I put in quotes, healthy sequence, but you can also put in a sequence that has something like a stop codon inside of it. And so therefore it would knock out the gene in question. And actually that's what this investigation is all about. It's one of the cool factors in it. And so once CRISPR system has made the cut, the DNA template right there can be paired up with the cut ends, recombining and replacing the original sequence that was cut with a new version. So it's bringing in a new piece. And so this is known as homology-directed repair. Pretty interesting, right? So depending on what the researcher is doing, they can design this donor sequence to whatever they want. As I was saying before, they could swap out nucleotides, they can add nucleotides, they can make the gene non-functional, they can insert a stop codon. There's a lot of options here. It's pretty cool. So how it actually is able to fit in, and thus the name, is that the donor DNA has on the outside of it, you can see here in blue, it has these homology arms, and those are homologous to the cut area. And so this is why you're going to be able to add this donor DNA, and it's going to be able to anneal and ligate in perfectly where the target was. And so we could use bacterial repair enzymes or machinery to actually stitch this in. And so, right, you can see that the donor DNA is going to go right into the area that was cut, right in there. And so that, again, that's homology-directed repair. And so to review this, so you start off by cutting it, so that the gene in interest, in this case LacZ, is cut by the single guide RNA that locates the sequence, and then the endonuclease Cas9 cuts it, and then the repair process is that this donor DNA is going to come in right at the cut region, and then look at this, and a stop codon 1, 2, 3 has been engineered into the donor DNA. And so when that's incorporated through homology-directed repair, the stop codon, if a stop codon is added to the LacZ gene, that should make it non-functional. And to remind you, if the gene is non-functional because of an inserted stop codon, then it's going to be knocked out, and therefore the E. coli would appear phenotypically white, since they won't be able to produce B-gal and therefore break down X-gal. And so in this investigation, the bacteria that's being used is HB101 plasmid BRKNAN. And so this is a particular type of E. coli that has been previously engineered, which I find pretty cool. It's been previously engineered to continuously express the Cas9 endonuclease. So that's a given. So Cas9 is going to always be expressed in this particular E. coli. So the kanamycin is a antibiotic, and so this particular bacteria was going to be grown on with this antibiotic, and so basically it's going to filter out any contaminant. And so it's also a selective marker for this particular E. coli strain. So it's going to be grown on kanamycin plates. And so the repair enzyme necessary in homology-directed repair is going to be needed to insert the donor DNA into the chromosome. But those sequences of DNA that code for those repair enzymes are going to be found on a plasmid. So the ability to repair the cut is going to be provided with a plasmid via transformation. And so that's what this investigation is about. It's not only providing the donor DNA, but it's also providing the repair enzymes necessary to conduct the HDR. And so that plasmid that provides the repair enzymes is under the control of the sugar Arabinose. And so there's an Arabinose promoter that is attached in the plasmid. And so if you're familiar with what I was talking to you before about IPTG being an inducer molecule that induces or provides the ability to remove the repressor protein for RNA polymerase, the sugar Arabinose will also, if the sugar Arabinose is provided, it will then allow RNA polymerase to easily transcribe and translate the inserted plasmid, providing the repair machinery. So if transformation is conducted, then the bacteria grown on Arabinose will be able to produce the enzymes necessary for repair. If you were to grow the E. coli on a plate that did not contain Arabinose, then they wouldn't be able to read the plasmid. Therefore, they would not be able to produce the repair machinery. So that would be, in a sense, turned off. And if they couldn't repair, you can think about this, if they couldn't produce the repair enzymes, then what would be the consequences of that? So the starter plate has two types of starter plates. The starter plate could either have Arabinose on it or not have Arabinose on it. And I'll say this, I don't want to reveal the possible results of the experiment because it truly is a discovery and an exploration. But I'll give you this, that when Arabinose is present, then that induces the plasmid upon transformation to actually transcribe and translate the machinery or repair enzymes necessary to insert the donor DNA into the cut region in the LacZ gene. If there is no Arabinose present, therefore it won't be read, and therefore the repair mechanism is turned off. Okay, so to review, the starter plates will either have Arabinose or not. Recall that all the starter plates are going to have IPTG, which is turns on the LacZ gene, and they'll also have xgal in them as a reporter. And the plates will also have the antibiotic kanamycin to select for those particular strain of E. coli that we're studying in this investigation. So the plasmid, in case you're wondering, also carries the kanamycin resistant gene, and so that's useful. So the Arabinose induces the homology directed repair mechanism because that's found on the plasmid that is part of the transformation. So Arabinose turns on the repair. So let's talk about these plasmids. There's going to be two types of plasmids involved in the investigation. I hope you're getting excited about this. So there's two types of plasmids. One of them is called the plasmid donor guide, which means that it not only has the donor DNA, right in here with the stop codon, but it also has the single uh, guided RNA, uh, the single guided RNA. So it has the guide and the template DNA. This one, just the PD plasmid, has the donor DNA. So they both have donor DNA, but this one doesn't have the guide as well. And so that's kind of an interesting consideration as well. So what would be the consequences of transforming E. coli and providing all of this, but not having the single guided RNA? And so plasmids, just to review, the plasmid PDG has donor DNA, so does the other one. Both have donor DNA with the stop code on it, but only the guide PDG plasmid will contain the single guided RNA uh, segment, which will then form a complex with Cas9. Okay, so the transformation itself is fairly straightforward, and so that's what I want to talk about in the remaining of this video. Is that the starter plate is grown on X-gal plates, and then plasmid is added using the classic heat shock transformation. Cells are provided with a transformation solution, uh, often magnesium chloride, and then they're chilled and then heat shocked and then chilled back on ice, and then they recover for a certain amount of time, and then they're plated on petri dishes to see phenotypic outcomes. Now, it should also be noted that these investigation plates are going to contain another antibiotic called streptomycin that's going to select for bacteria that have been successfully transformed. Both of the plasmids contain resistance to this particular antibiotic as well. So this is a good summary diagram here. So the E. coli that are disrupted by CRISPR, in other words, have been cut, will have a non-functioning LacZ gene. Okay, so that means that they'll be white if they're repaired. So, and I didn't mean to laugh, but it's kind of this is crucial, because if repaired, because if it's not repaired, sometimes double-stranded cuts will result in the death of E. coli. So that's a possibility. So that needs to be considered. And so, here's your starter plates, one with Arabinose, one with not. As you can see here, this one has Arabinose, this one does not have Arabinose. And so if it does not have Arabinose, that means it doesn't have the repair enzymes necessary. If it is grown with Arabinose, then it will be able to do this. And so both plasmids contain donor DNA, and the PDG contains the single guided RNA. And so these are going to be plated, as you can see here, on 4 petri dishes, A, B, C, and D. And so A and C are going to have a plasmid donor, so that's going to have the plasmid, but no single guided RNA. And plasmid donor guide has both, so that's B and D. And then the colonies being transferred to A and B will be from the petri dish that has no Arabinose, and then C and D will be transformed from the petri dish that has Arabinose. So there's kind of a lot going on, which makes it intriguing. I like that. And so just to review this, the bacteria will be grown in 2 different starter plates, one with Arabinose, one with not. All the bacteria are producing the endonuclease Cas9. Bacteria that are not growing on Arabinose are not going to have the repair mechanism. Those that are growing on Arabinose will be able to have the plasmid reveal the repair mechanism necessary to ligate in the donor DNA. And so the transformation, in other words, getting these plasmids into the E. coli, is pretty straightforward if you're familiar with this. First, you have to make your starter plates, and then you have to start up. Then you have to do a starter E. coli, streak your E. coli, and incubate overnight in 37 degree. Again, there's going to be two types of starter plates, one with and without Arabinose. And so these are going to be grown up for a day in an incubator, or a few days at room temperature if you don't have a 37-degree incubator. And then basically transformation solution is added to all three microtubes. You can label them A, B, C, D, 250 microliters, and then chilled. And here they are chilling. And then you're going to transfer from the starter plate about approximately 5 colonies by using a sterile inoculating loop and scraping them up and swirling them in to each of those. Both, again, A and B are coming from the non-Arabinose, and C and D are coming from the Arabinose plates, 5 colonies that are transferred in there. And once the cells are mixed with the transformation solution, they're chilled on ice for about 10 minutes. And then the plasmids are added. And so 10 microliters of plasmid donor to A, 10 microliters of plasmid donor to C, plasmid donor guide goes into B, and plasmid donor guide goes into D. And then, of course, the heat shock is crucial. It works optimally at 60 degrees Celsius for 50 seconds, no more, no less, and then quickly place back on ice for 2 minutes. And then after 2 minutes of ice, you take it off and put it at room temperature, and you add 250 microliters of LB broth, which is their favorite recovery beverage, and let them do their thing for at least 20 minutes. This is a possible, if you're conducting that, this is a possible stop point. You could let them sit at room temperature with their recovery broth overnight, and they'll be fine. But if there's time in your day, you can continue right through. And so what you'd want to do after 20 minutes of recovery is then transfer those cells 100 microliters using a P200 micropipetter, and streak them on your A, B, C, and D plates. Okay, so that's pretty straightforward. And then, of course, they grow best inverted, condensation wise. You place them in the 37-degree incubator in the dark. And those particular plates, you want to think about it. I mean, you could pause this and contemplate that and really try to figure out, well, what would A look like? What would B look like? What would C, and what would D? And the cool factor here is that, you know, if it's really important that you do take the time to think about this and predict possible outcome, because though it may be a challenge to you, it'll cause you to really think and make personal things that I've been saying so far. And so the expected results, and I'm not going to tell you what the actual results are, but the expected results would be things like no growth. That's a possibility, right? There could be colonies growing, and they could be white. You know, what's the story there? Again, no growth. And then there could be colonies, and they're blue. And so what each of these are trying to illuminate is the various components of CRISPR-Cas9 cut and repair. So it's trying to inform us of all the things that we've been discussing so far. And so hopefully you'll consider conducting such an experiment, and I think you'll find it rewarding. And again, thanks to friends and colleagues at Biorad Laboratories for designing this particular investigation. And thank you for watching.